is required is to have an understanding of the binding kinetics and binding affinities in SPR. So uh, which does not depend on any kind of fluorescence tags or fluorescence levels, but it depends on the biomolecular interactions. So it takes in account of the responses and for changes in responses according to the change in the diffractive index. But the major important thing is to have a very good idea about the surface chemistry and the different kinds of sensors available, so that to make it very much fruitful and to design a successful experiment. So the again SPR, what advantages it gives us is that first of all it is a level free measurement, and it gives us analysis in real time, and can be done on different crude samples, and it is one of those where we can have the ligand analyte binding. So the major thing is that once we get the once we get the chips where the lights are reflected, so the major thing is that the surface in the surface where the ligands, so all the which is immobilized on a chip is known as ligands, uh, and the analyte whatever it is flow to find out the binding. So these analytes are those which gets bind to this ligand, and one can understand the changes what happens. So now to whatever major thing that we will be today looking upon in these weeks, which I hope that you all have gone through, is that what is the surface chemistry, which type of interactions to be looked upon on which kind of chips, and to how to uh, have an understanding of that, as well as how to understand the different steps associated with it, like the sample preparation, surface immobilization, then the regeneration steps, yeah, yeah, and so all these steps, what we will be looking upon. So the questions that can be answered for surface plasmon resonance is that if it binding, yes or no, the specificity, affinity, kinetics, concentration, etc., etc. So this gives us a better understanding of the molecular binding and the biological functions, and ranges as it ranges from monitoring events between molecules, from ions to viruses. So all those things can be monitored by either different kinds of ions, different kinds of small molecules, bacteria, viruses, etc. That different varied range of applications, and that's why it makes surface plasmon resonance a very kind of a robust technique for varied applications in finding different kinds of biomolecular interactions. But also we need to understand that this kind of biomolecular interactions, that what actually happens, so like the nucleic acid, protein, or lipids, etc. But the surface chemistries are different. Chemistries are different for interaction of proteins or nucleic acids or lipids on the different surfaces. So it becomes quite important for us to understand that which kind of biomolecule will be actually binding to that surface, can be mobilized further. Different analytes can be uh, given forward. So now if we see that whatever in these steps, if we see the first it is a typical sensogram which shows us the different units, like it is the baseline, then the association, then the dissociation, then the regeneration steps, and this is the where the injection. So again, one major thing is to have a very robust kind of injection, which shows us the way that how uh, the association will depend and what we looking into it, different kinetics and binding. So analyte injection, then the immobilization and the regeneration steps is again reusing the chip and making the surface available for the next set of analytes. So this is are some of those important steps that we need to keep in mind. And this is a typical sensogram where responsiveness we get on the basis of the time. So what are the information that we can generally retrieve from surface plasmon resonance? So this is that first thing is that it analyzes molecular interactions in real time and obtain a wide range of clinical binding related data. So you know that the binding related data is quite important and has also different applications. So for example, if you can understand that which epitopes will be binding to your antibodies or different kinds of generation programs in kinds of that binding interaction is strong of the epitopes and the immunogenicity for elucidating for making manufacturing different kinds of vaccines, etc. So all these binding related data are quite important and can also be used for the interaction. So for example, if you we all know that the virus COVID got attached to the S2 proteins of the receptors of the human. So which particular protein is responsible or associated for having for binding such kind of data are quite critical and understanding are quite important. So this helps us to have analyzing in the real that time. So for example, if we find out that which particular type of uh, if the mutations happens in certain bacteria and viruses and it is getting attached to which protein of the human, so this kind of interactions we can easily get. Also if you design certain kind of drug and to find out the physical interactions between your protein and drug, if you find out your physical interactions protein and drug, you can easily do so and you can find out the if it is binding or not and what is the specificity. So it is able to detect, identify specific binding partners and characterize the binding like affinity, kinetics, concentration, etc. And as I mentioned that it can analyze like the nucleic acids, lipids, carbohydrates, cells. So also the membranes for the lipid which are getting attached to it. Uh, which uh, which gets the lipids and interacts with which kind of molecules for getting it into entry into the cells and etc. can be identified from this kind of thing. So now, what is the advantages of SPR and what is the design? That it is a microfluidics based design. So it takes into the principles of microfluidics where the fluids and the concepts. Second is that miniaturized system. The system is quite miniaturized. So we will also look what one example where we can see that this can be used in uh, sorry, we see that this portable portable or miniaturized. So if I say that the portable miniaturized system, whatever we get, these are very much useful in clinical applications because with large machines, and if you cannot move across, if it is not portable, or if it is, if at all, not also handy, so then what you are doing is that you are saying that it is not so much useful or not so much taken forward. But SPR has this kind of design with a miniaturized system, and it can be taken forward for the clinical setups as well. And we will look at the example at the end of this thing, that where they have people have reported of using this kind of portable for different clinical applications, which can be a very much suitable thing. Second, third is the low volume of the reagents and integrated and the automated liquid handling. So from the analyte flow and uh, all those things can be taken, are incorporated in the design for better reproducibility, because if you do the volume and all those manually, then definitely there's a chance of introducing variability. Whereas if you do it through integrated and automated liquid handling, it does not uh, lead you to the any changes. So this is one thing which help us in uh, understanding of those things. So now is that surface preparation, or how to immobilize the surface. So the major thing is that uh, one of the major importance is to have the immobilize your surface, means that you need to immobilize your so what does it means? So you have the surface. So now you need to place, immobilize your ligands, right? So if your ligands are placed, for example, your ligands, this is your ligand. And this is your analyte, right? So this is your ligand and this is analyte. Now the ligand needs to be immobilized onto the surface, so that the ligand is immobilized onto the surface, so that whatever we are getting, that analyte should get fixed to it, so the ligand should be there. So now the one thing is that direct immobilization, which we have the covalent chemistry and have higher binding capacity. Example is amine ligand, thiol, surface thiol, melanimide, aldehyde, etc. Second is the capturing or the orientation specific, which has the lower binding capacity, like the streptavidin, biotin. So these kind are two uh, kinds of things. One is the direct immobilization. Second is the capturing. So one is orientation specific, and another is using the covalent chemistry, which has a higher binding capacity. So now, what are the different kinds of sensor chips? So now I think you all have understood that one thing which you need to have the uh, immobilize the ligands, and one is to have the covalent bonding of the direct mobilization using covalent chemistry or using the orientation specific, which includes your like the spectrometry, binding, etc. So now the sensor chips, among which one is Tristan CM5. So it contains a matrix of dextran. This is what it is known as is one kind of the most kind of versatile chips and have possess excellent chemical stability and assay. Right, so this contains dextran, carboxymethylated dextran matrix, and it is kind of a versatile chip, and it is having possesses an excellent chemical stability and assay. So what it is? It contains a dextran matrix. So it is hydrophilic and it is flexible and it resembles a 2% aqueous dextran solution environment. So this chip, what it is there. So what any amount of your uh, this dextran represents your 2 2. So if you make 2% aqueous, so this is your 2% aqueous dextran solution. What it resembles if you make 2% dextran aqueous dextran solution, so it is resembling those. So the major advantage of it is, which we have said, that it possesses a very high stability. So the thing is that it also ensures low non-specific binding. So one thing which we need to be careful of if we try to find out and report any kind of uh, binding, we should ensure that it is low non-specific yeah. So if we say that it, uh, second is that it is easy to activate and is also used for covalent coupling. So the major thing that it is being used, one thing is that whenever any kind of surface is there, and we need to immobilize and we to immobilize ligands, we need to activate the surface as well. So this one matrix for the sensor chip and also it can be used for covalent coupling. And finally, the important another thing is that it has also can be used for extensive uh, regeneration. So first chemistry, what we can understand that it is hydrophilic and it is resembling the aqueous Western solution, and it uh, it ensures a low non-specific binding, and it is also easy to activate and use for covalent coupling, and it is used for extensive regeneration. So this is one of the chips for the CM5, which is been very much used. So most also, so this is also regularly used, this chip. Okay, so this is the what thing is that it which has to have a low non-specific binding and easy to activate for ligand immobilization. Now next chip is sensor chip, which is known as CM4, which is having a lower degree of carboxylation than TM5, which is for which is that which less negatively charged. So one is CM5, which is of the dextrans chip with carboxylation with a higher degree of carboxylation than CM5. Another is for the CM4, which has a certain state of lower kind of carboxylation than CM5, less negatively charged. So it reduces again non-specific binding of highly positively charged molecules as found in of highly positively charged molecules as found in the cell culture supernatant or cell origins for basic filtering. And it is also convenient for load aromas. So why less negatively charged is beneficial? So it's why and when. reduces the non-specific binding of those highly positively charged molecules. Which are found in the cell culture supernatants and different cell homogeneous. So for the basic protein, so that's why if it is less negatively charged, positively charged molecules, whatever highly positively charged molecules that are generally found in different cell cultures and supernatants and cell lysates, so that would be less attracted. So So this in a way will be helpful because it is having a lower negatively charged surface. So those highly positively charged won't be binding to it and will ensure less kind of non-specific binding. And it is also convenient for that low mass. So till now what we have, so we will also do a summary of the different kinds of chips that we go through. So till now we have done for the CM5, carboxy methylated So which is the hydrophilic one, but the major thing is that it can be easily active regeneration and easy to activate. So actively it can be regenerated. Uh, okay, but the thing was that, and it can also be get uh, activated quickly. But the next was which we had talked about is the CM4, which is just less less carboxymethylated, and which is having a less negatively charged and can be used for reducing non-specific binding, non-space binding because of different highly positively charged molecules that generally comes on the cell surfaces of cells. So now this is sensor chip C1, which is having a flat carboxymethylated surface. So this is flat carboxymethylated, and it is used for the positive charged proteins, and the dextran matrix is not required. So this is the simple carboxymethylated surface. So till now what we had seen in CM5 and the CM4, it is the carboxymethylated surface with the uh, dextran, but here it is one which is the flat carboxy methylated surface without any kind of dextran matrix is not required, and is used for the positively charged proteins. Okay, and the dextran matrix is not required. Now the next chip is the sensor chip, uh, uh, which is say again this is a dextran matrix but pre-immobilized with streptavidin. So now it captures such as carbohydrate, proteins, and uh, DNA, which captures the biotin ligands. So now, which we had said that there are two kinds of immobilization. Which is direct immobilization and depends on the and it depends on chemistry. Another was the orientation specific. Is the orientation specific. So the carboxymethylated dextran matrix that is pre-immobilized with streptavidin. So which one is pre-immobilized with the streptavidin, and it captures the biotin ligands and with carbohydrates, peptides, proteins, and DNA, and it is ideal for capture of large biotin DNA fragments and for the study of different nucleic acid interactions. So till now, what we have seen that one, if we just till now we have seen CM5, which is robust used and is carboxymethylated dextran tubes, which is hydrophilic ones, and are used as an hydrophilic uh, ones and is used very much, which easy to activate and activity generation. Then we have CM4, so which is less negatively charged and less carboxymethylated than CM5, but and used for reducing the non-specific bindings. Then we had one to C1 where large positively charged proteins and where dextran matrix is not used, and now SA sensor chip SA, which is a kind of a thing where the so it is carboxymethylated dextran matrix, but it is immobilized with the streptavidin so that biotin ligands such as carbohydrates, peptides, proteins, and DNA gets interact to it. So it is actually ideal for capture of the large biotin DNA fragments and study of nucleic acid interactions. So next is the sensor chip of NTA. So which is again carboxymethylated dextran matrix, which is pre-immobilized with NTA. So this is the so which is pre-immobilized with NTA, so which captures the his tagged ligands via metal chelation. So it captures via metal chelation. and this controls the more optimal site exposure. So the major advantage is that it controls the steric orientation and ligand component for optimal site exposure. So now CM5 CM4 C1 is a SA and NT, right? So CM5 and NT. So among which it is binded with the stability for the capture using the biogenerated ligands, but to orientation specific immobilization, and the NTA which captures your his tag ligands via metal chelation. So this is quite uh, important in that way. Now next is that sensor chip HPA, which is again flat hydrophobic surface, and it is alternative to the solubilization techniques for studying membrane associated interactions. So it is different for the solubilization techniques for studying the membrane associated interaction. So as I have already mentioned in the start, that the membrane associated interactions are also quite can be well studied using SPR or the surface placement resonance. So the sensor chip HP is one which gives us the opportunity to study using a flat hydrophobic surface, and it is alternative to the solubilization techniques for studying the membrane associated interactions. So the receptors occurred in membrane-like environments and interacts with the analytes in the aqueous buffer. So the receptors which are there and anchored in the membrane like environments and interacts with the analytes in the aqueous buffer. Now the second thing is that, so this is the lipid monolayer interacting with the membrane binding biomolecules. So the So the sensor chip HPA that is gives a hydrophobic surface and it is for the membrane associated interaction. So the environment mimicking the environments for the membrane-like environments with the aqueous solution, aqueous buffer, and interacting with the binding molecules. Last is a sensor chip AU, which is having a kind of an unique surface chemistries and the plain cold surface. So if I just again give you a brief, CM5 most versatility used with having a very good stability and reproducibility and is being useful for this kind of thing. But the major thing is that this CM5 is at carboxymethylated strand. Next is the CM4, which is less carboxylated, less negativity charged. Then it was C1 without the dextran matrix. So what was its benefit or advantage? That the highly positively charged substances which comes in the cell lysates and all will not get bind to it, and it will reduce the non-specific bindings. Third is the C1. C1 which is a flat surface without having any. So this flat surface without having any dextran matrix for highly positively charged proteins. Then we had after C1 we have SA. So this was for the immobilized with streptavidin, the carboxymethylated dextran matrix, immobilized okay, so immobilized with the streptavidin. So this is immobilized. So this gets immobilized with the streptavidin. So the different bioactivated ligands like your DNA, carbohydrates, peptides, proteins, and DNA gets bind to it, and it is for capturing those, immobilizing those DNA fragments, and then studying for this kind of different nucleic acid interactions or other interactions. Similarly, for the NTA, is that it is immobilized with the NTA and can be used for the histag ligands with the metal solution, and it controls the steric orientation for optimal site exposure. Correct. So now, if we move to HPA, which is flat hydrophobic surface, and it is for the membrane-like interactions, which mimics the receptors and protein membranes like environments interact with the analytes in the aqueous vapor, and the lipid monolayers interacting with the membrane binding uh, biomolecules. So, and last but not the least is a NTA, nickel NTA bind for methylation for histag ligands. And then finally is the HPA, which for the membrane hydrophobic, flat hydrophobic surface. For membrane associated interactions. And last is the lipid monolayers interacting with the membrane binding biomolecules, and then say AU with the plain gold surface, which has have a very unique kind of surface chemistries. Now, what is the experimental design or the place? One is that the analyte ligand. So again, to repeat, that you have this, where you have the ligand, analyte gets bind to it. So this is the analyte ligand. The general steps is the surface preparation. So first step, which we have repeated a number of times, is that the surface preparation. So this chip surface is prepared Okay, so the chip surface is to be prepared. So this is the by immobilizing the covalent linking of a ligand or capture of a molecule to the sensor surface. So now the choice of immobilization strategy depend on the ligand properties, as I was saying, that the bioactivated ligands, so you need to have a chief with the streptavidin or assay, or CM5, CM4, which is having hydrophilic and having less positively charged, and all those things which needs to be keep in mind. So this is strategy will be completely dependent on the ligand properties. Now the sample injection can be of different types: quick uh, there's a two major concept of one is the binding effect and another is the bulk effect. One is the binding effect. One is the binding effect and one other is the bulk effect. So if you say the bulk effects are due to the differences in the refractive index of running buffer and sample preparation. So that is so due to the difference in the running buffer and sample preparation. So if when you are preparing the samples, so now this gives us to another hint of sample preparation and the running buffer that we are doing. So if their refractive indices are different, the sample preparation and running buffer, their refractive indices if their refractive indices are different, then there will be some effect because there will be some changes that are associated with it. So now, why refractive indices again? Because if we again go back to the basics, that the SPR, the response which we are getting with the changes in the refractive indices because of the binding, what is happening now with the sample preparation as well as whatever we are preparing. You might say that we are preparing the running buffer which we are giving during the injection steps. If those refractive indices are different, there might be some differences and some effects. So this does not reflect to the true binding to the surface. So this is one of the effects which we need to keep in mind whenever we are trying to design this kind of experiments and we are reporting any such kind of which reflects the true methodology of binding. Next is the reference surfaces. So if the sample matrix and the running buffer differ, so in some situation, not if at all that be the case where you are having the sample matrix where you have the sample matrix and the running buffer differ, if both of them finally leads you to the differs and it differs, so then what you are saying that the bulk contribution can be subtracted using a difference bulk. So this bulk contribution or the bulk effect, which caters to or hinders to understanding of the true binding, can be subtracted by using a reference bulk. So the major problem that one faces when this kind of happens is that uh, we don't have the reference for, and we don't have the true understanding of the changes of the bulk effect due to the difference in the sample matrix and the running buffer. So this contribution can be subtracted by using a reference bulk, and it should be placed upstream of the active surface. So design of reference of surfaces can be like the unmodified surface, activated, deactivated surface, immobilized vitamin against sample considerations, homogeneous samples, solubility, specific additives, etc., etc. So these are the some of the changes that we need to uh, keep in mind. Okay, so this is the design for the reference surfaces. So this problem is that the bulk effective, the refractive index of the sample matrix and the running buffer differs to some extent. Then what? So they what had happens is that the bulk contribution can be subtracted using the reference bulk, and it should be placed upstream of the active surface. So the majority is the design of the reference surfaces, from which the bulk effect can be reduced and can be subtracted. So unmodified surface, activated, deactivated surface immobilized with dummy ligand, and so the sample considerations, that is the homogeneous samples, and buffer requirements. So must be filtered and degassed or deionized, and solubility and that is specific additives. Now another uh, step is the regeneration step, which at the, if I remember, if I when I had shown you the complete, so this is the regeneration step where you need to regenerate the or reclaim the surface of the sensor, where you want to remove all the bound analytes from the ligands. So that step is also quite important. Uh, so one thing is that removes the bound analyte completely from your surface, and activity of the surface must remain unaffected, and there should be no change in the activity, and the efficient regeneration is crucial for high quality data. We should have a efficient regeneration process in step, which is crucial for the high quality data, for high quality data. So this kind of regeneration is quite crucial and quite important need. So it removes the bound analyte, which is completely from the surface, and the activity of the surface must remain the unaffected, and efficient regeneration is crucial for the high quality data. So you can understand, this is a plate with the analyte. This is the ligand. Analyte. So regeneration steps includes removal of analyte completely from the surface. So the activity of the surface that should remain unaffected. Activity this should remain unaffected. So the activity should not go. So this should not be so harsh that the ligand gets completely washed off. Rather than the only the analyte is getting washed off, and this is crucial for the high quality data because the binding and the affinity what we try to find out depends on a lot on this kind of thing. So this is very much important. So this removes the bound analyte completely from the surface, and the activity of the surfaces must remain unaffected, and the efficient regeneration is crucial for this high quality data. So this is one important. So you can visualize that you have the ligand, you have the analyte bound to it. So you are removing the analyte without affecting the activity of the surface, and this is crucial for the high quality data. So this is how a typical SPR thing is. So you can see. So what are the things that we were mentioning, that this is a glass, metal, or plastic based gold layer. This is the ligands. This is the immobilized matrix. So matrix immobilization, that's what we are saying. And this is the analytes. So which you can see that it is getting bind to the our ligands. So just now visualize which one is the ligand and which one is your analyte. So this, so analyte is something getting bound to your so this is what means, and regeneration state means completely removal of ligand. So this is that complete removal of analyte. Complete removal of analyte in the regeneration step for it the surface to again be free of the next set of analytes to be get added. So this is quite important step, starting from the immobilization to the regeneration steps. So now again, just giving you a brief of whatever sensors we have read upon. So the carboxyl amine, gold, biotin, NTA, proteinase sensor kit, liposome, hydrophobic, all these are there apart from what we have read, like the carboxymethylated dextran, CM5, CM4, C1, SA, NTA, HPA, gold. So proteinase sensor kit, liposome, hydrophobic, NTA, biotin, streptavidin sensor kit, gold, amine, carboxyl. So all these are some of those sensors that can be used and utilized for SPR. So this is our short notes of what we have already discussed of the carboxymethylated esters. So if I just read it out for you. So CM3 or short dextran is that sensor chip CM3 provides the same functionality as sensor chip CM5 but has a shorter carboxymethylated dextran matrix. A shorter matrix may be of value in work with large analytes such as cells and virus particles. It may also give reduced non-specific binding in work with serum and can be valuable in testing the influence of the CM5 Western matrix on kinetic determinations. Experimental protocols follow the same principles as applied to sensor chip CM5. Due to the shorter matrix, the immobilization yield will be approximately 30% of that obtained on sensor chip CM5 under comparable conditions. CM4 is a dextran mixed with chip CM5 but with a low degree of carboxylation. So which we have already discussed. It provides the same functionality as sensor chip CM5 but is less negatively charged, and this lower degree of carboxylation may be of value for reducing in non-specific binding in what we analyze as a high positive charge or group samples such as cell culture supernatant and cell homogenates. And sensor chip CM4 may also be useful in kinetic applications where low ligand densities are important. Experimental protocols follow the same principles, but due to lower degree of the immobilized yield will be approximately 30% of that obtained of sensor chip CM5 under comparable conditions. CM5, that is standard surface sensor, is a general purpose chip. So this is one of a kind of a general purpose chip that is being used for interaction analysis involving all types of biomolecules such as small organic molecules, proteins, lipids, carbohydrates, and nucleic acids. A sensor chip designed to allow detailed quantitative studies of this regards field data, example interaction kinetics, affinity, concentration, binding ratios, covalently bound to the sensor via carboxyl moiety of the dextran, and which is used as on a ligand that can be used for couple includes NH2SH CH1 COH. Is regenerated by selective dissociation of the analyte from the covalently immobilized ligand. Conditions should be chosen to achieve complete dissociation of the analyte without affecting the binding characteristics. And CM7 is something which is known as your high carboxylation. Higher density attached to the addition, the degree of carbons are higher will give the sensor approximately 3 times higher immobilization capacity. Molecules are covalently coupled to the sensor surface via amine, thiol, aldehyde, or carboxyl groups. This surface is ideal for applications which use small molecules and fragments, and less suitable when working with the large biomolecule analytes, and it is very much less suitable with the working with the large biomolecular analytes. So it cannot use large molecule higher methylation. That's why the short low carboxylation and CM4 and CM3 we have found out and have been used. So we cannot go for the higher carboxylation, and we should not go for when we are using the large analytes and those things. So now from the SPR signal to the sensogram. So how to make sense of this kind of data? So which we will be now looking upon. So just look into the standard experimental design and setup. So as we have already spoken, like this is the gold Now you have the prism. So this is a light. You have a protein and you have an antibody, right? So antibodies in the bulk solution governed by mass transport approaching surface immobilized proteins by diffusion. So it will, by diffusion, by the process of diffusion, it will approach to the surface immobilized. Okay, now what it is known as the studies or as the concept from the SPR signal to sensogram, which we can say is that in an SPR experiment, a deep or minimum in the reflectance spectrum signifies the angular at which the light has been absorbed by the surface plasmons. So you know that the plasmons are some those which gets absorbed and excited. Once a change in the refractive index at the sensing layer caused by bound molecules causing surface structural changes, the deep would shift, indicating a change in the angular wavelength of resonance at which the plasmons absorb and resonate. Basis of that biomolecules, when it gets added, certain weights and certain masses gets added to it, then definitely what it happens is that it changes, causing a structural surface change, indicating a change in angular wavelength of the resonance. So now reflecting spectra with different RI on an AU sphere sensor, so increasing refractive index. Now what it is happening, the detected change or wavelength is proportional to the amount of the newly binds. For example, in the number 1 position, it's in the baseline in the number 1 position that you have just immobilized your antibodies on a chip. So in the is proportional to the amount of newly bound analytes at the surface due to an increase in mass. So then there is a increase in mass and there is a change. Response is plotted with time a sensogram is produced. Sensor is proportional to the amount of bound analytes. On and off rates, K on is association, K off is the dissociation, Kd is equal to K off by K on. Baseline, so number 1 is baseline, 2 is association, 3 is steady state, 4th is dissociation. Steady state means that there is no increase in the changes in the refractive index, though there is a change in the analytes. Kd is the dissociation, regeneration, and Ru represents the response units, resonance units. So now a sensogram is composed of 5 phases: baseline, association, steady state, dissociation, and regeneration. So please keep in mind that a sensogram of what we are talking about of surface plasmon resonance. So this technique is very much clear from its literal meaning of the name that it's provided, that is surface plasmon resonance. So at the surface, whatever plasmons are there, it's absorbed and it resonates to provide you the energy, to provide you the uh, unit or the refractive index. So this comprises of baseline, association, steady state, dissociation, and the regeneration. So initial phase is a baseline. A running or flow buffer is used. So there is a running or flow buffer which is used to condition the sensor surface. Sensor surface gets conditioned and check for any sensor system instability. It is crucial to have a flat baseline because any drift, injection, spike, and high buffer response is an indication that the system should be checked and cleaned. Standard running buffers include phosphate buffer, saline, and HEPS, NaCl, etc. So such as standard running buffers. So in that initial phase, a running or flow buffer is used and condition the sensor surface. So whatever sensor surface is conditioned and check for any sensor system instability, that any spike is there or drift are there or high buffer response rate, that the system should be checked and cleaned. And the standard running buffers include VS and HIPS NaCl. The second phase is association, where analytes begin to bind to immobilized ligands. So again, surface immobilization is one of the major important things. It is indicated by the initial sharp rise of the SPR signal in the sensogram, and it is ideally a single exponential curve. The steady state occurs at the top flat portion of the sensogram, where the net rate of bound analytes is zero. Means as many are getting associated, as many are getting dissociated. So that is the steady state is zero. A dissociation phase begins when the analyte solution is replaced by a wash buffer, which causes specific bonds between the analytes and the ligands to break. It is represented by the downward slope after steady state phase. Regeneration phase, consisting float of pH buffers such as glycine, in order to obtain the same SPR baseline signal as the beginning of the experiment. Just move out the analytes. Steady baseline, that the sensors are free of bound analytes and non-specifically absorbed molecules, has stability, having intact and functional receptors, that no other effects such as temperature or surface chemistry changes would affect the next set of SPR measurement, as SPR sensing surfaces can be reused many times. Okay, so but before going to that, we will just look into here. Okay, so this gives you complete clear idea of how it is given. So it is a plot of SPR response versus time generated from an SPR instrument, whether there is a binding event, whether the binding is specific. It contains valuable kinetic, affinity, and concentration information pertaining to the analyte and binding of interest. So the analyte is a biomolecular molecule being investigated. So analyte, so don't get confused between the ligand and the analyte. The ligand is something that is being immobilized on the surface, chip surface. An analyte is something that is going to bind and to or the molecule which is being under investigation or under study. So the analyte is the biomolecular molecular being invested, and the ligand is the recognition element being immobilized on the SPR sensor. So this how sensogram is obtained, how it is generated. A deep or minimum in the reflectance spectrum, intensity of reflection versus angular wavelength. The intensity of the reflection signifies the angle at which the light has been absorbed by the surface plasma. Once there is a change in the refractive index, shift, you can see there is a shift. So typical sensogram, which consists of on and off rates, on and off rates, various parts of the sensogram. So once the sensogram data are obtained, the rate of association, that is K on, dissociation, that is K off, and dissociation constant, that is Kd, can be determined by fitting the data into a suitable binding model. Therefore, kinetic and affinity data can be obtained readily from an SPR experiment or surface plasma response experiment. So then it's generating a sensogram. So with wavelength interrogation mode, the detector detects a change in the wavelength of the absorption band minimum or deep upon the introduction of a sample, which could change the refractive index near the sensing layer. As the sample continues to flow across the sensing surface, as it is continuing to flow across or sensing surface. Across the sensing surface, the wavelength position is continuously detected. And recorded in the SPR instrument in real time. So how it is done? So as the sample continues to flow across your sensing surface, the wavelength position is continuously detected and recorded. Okay, so the wavelength position that uh, of the change resulting is a plot of delta wavelength referenced against a solution at a specific time versus time. So delta wavelength of the change in the wavelength. Fitting the sensor into suitable binding model as mentioned above. So are already integrated. The change in the shift of the wavelength of the index specific to the samples, and then this shift is minded to a data, raw data, and the SPR shift, change in the shift. Okay. So this is analysis of protein heparin interactions using a portable SPR instrument. So which I had already told that we will be showing one example that how this kind of uh, technique or this kind of instruments can be used for a in clinical setup for different interactions to study. So optical biosensors such as those based on surface plasma resonance are a key analytical tool for understanding biomolecular interactions and function as well as a quantitative analysis of analytes in a wide variety of settings. The advent of portable SPR instruments enables analysis in the field. A critical step in method development is the passivation and functionalization of the sensor surface. Passivization and the functionalization. So we describe the assembly of surface of thylated olylethylene glycol, biotin olylethylene glycol with streptavidin and reducing and biotinated heparin for portable SPR instrument. The two examples, heparin binding proteins are presented, the binding of fibroblast growth factor 2 and comparison for the binding of heparin sulfate. Library of selectively modified heparins and sulfamines which identify the selectivity of the enzyme sugar receptor site. And heparin functional substances should have a wide applicability. Is a close structural analog of the host cell surface polysaccharide and receptor for many indigenous proteins and viruses. Other important thing or which is being used like the kinetics of protein-protein interactions at the surface of an optical biosensor, which has been so. So methods based on the use of optical biosensors have recently become available to provide a convenient means of determining the rate and equilibrium constants for biomolecular interactions between immobilized ligands and the soluble ligate molecules. However, the association data that these methods provide are not always accurately, and when the ligand is immobilized on a dextran matrix, we show that a better higher ligase activity and double exponential two rate limiting processes are involved. So they have talked about mostly the models, the how the data is picked. So now we'll see that. So this is of a guide which can help you whenever you are doing or running an SPR experiment. So how it can help you is that. So I'll just get through here. So the guide to running an experiment is that follow the startup procedures. So with the running buffer, prime the instrument and normalize the chip. So with the uncorrelated solution order operations, okay. So immobilize the ligand to a high surface density. So this is the first thing. So this say that the high surface density of 3000 to 5000 RU resonance units ensures that even if subsequent binding of the analyte to the surface is very weak, the binding signal should still be detectable on the noise and not bulk shift. So again, you can see the concept of bulk shift. That's why we had spoken of beforehand that what is a bulk shift and bulk effect and how it is done. That is a reference subtracted sensogram will give a signal 20 to 100 RU. For direct, again, the concept of the immobilization direct or the orientation specific ligand should that you prepare will vary for each protein. Generally, the ligand should be diluted around 5 to 20 microgram per ml in 10 millimolar sodium acetate buffer at the desired pH, that is pH 5. Activate the surface of contact over time. So we had already spoken of activation, of which CM5 was one of the major advantage was to that it can easily get activated. Uh, so for a 24-20 second or 5 micromolar is a recommended flow rate. Being the immobilizations with a 30s injection at 5 microliter per minute, the injection or contact time should be increased for subsequent injections as necessary to achieve the desired immobilization level. Little ligand is immobilized and during the first 30 seconds, increase the concentration reach the desired 5000 to within 5 to 10 minutes worth of injections. And then finally deactivate the surface. So verify your specific binding of an analyte to your surface. Really introduce two different concentrations of analyte, example 50 nanomolar and 200 nanomolar. So just think of how you will be designing the experiments and how you will do the troubleshooting and the cross check that whatever you are doing is fine and it is not getting changed. So verification of the binding can be easily accomplished by injecting two different concentrations of analyte. Example of is 50 nanomolar and 200 nanomolar, which is diluted in your running buffer at 30 microliter per minute and regenerating between each injection. Flow rate, so 30 to 50 microliter per minute. So this is your recommended flow rate for analyte binding dissociation and regeneration. If reliable specific binding is observed, proportional increase in binding signal with increasing concentration, 50 nanomolar to 200 nanomolar, if it is specifically binding to your ligand, the analyte. So if you increase your concentration, definitely it will get increased. Once ideal regeneration conditions are established, verify the binding signal remains stable for each analyte concentration. Now identify the ideal regeneration condition. Always work from the mildest regeneration conditions to the strongest. Avoid injecting regeneration solutions more than 60 seconds per injections. So these are some of the tips and tricks for setting up a successful SPR experiment. So what you can see that multiple short injections are preferable to a single long injection, which tend to damage the surface less. Condition should remove greater than 90% of the analyte bound to the surface. This can be determined by calculating the difference between the post-regeneration baseline and the pre-analyte injection baseline divided by the total analyte binding signal. The binding report point for the analyte injection, if the difference in baselines is less than 10% of the total bound, then uh, the greater than 90% surface regeneration has been achieved and the regeneration condition is identified. Once greater than 90% surface regeneration is achieved, inject the same concentration of analyte for the same amount of time to verify the regeneration conditions did not damage the surface. 16-second injection of 50 nanomolar analyte generated a binding of a signal of 200 RU, then an identical injection post generation should also generate a binding signal of about 200 RU, so for the same concentration. Exhibit a degree of non-specific population following the first one or two. If there is a difficulty, they try injecting the analyte and verify that a similar binding response is achieved. So baseline 0 RU, post 1 250 RU, post reduction 50 RU, 80%, 5 RU, 7 and 97%. Then is for the kinetic titration. At this point, reliable detection of the analyte to the surface immobilized ligands has been established and appropriate regeneration condition has been identified. High density surfaces are not suitable for obtaining the reliable kinetic energy due to the issue of the analyte binding. Lower surface density. So theoretical Rmax equals to molecular weight of analyte divided by molecular weight of ligand into immobilized ligand RU into stoichiometry. Number of analytes that bind to a single ligand. So for example, so this is an example which we can do. So protein analyte is 30 kilodalton, antibody is 150 kilodalton. So Rmax is 30,000 by 150,000 kilodaltons into 3,500 RU into 2. So theoretical Rmax one should get is 1400 resonance units, but observed is 1150. So that is 82%. So during analyte injection, so if a question is asked to you like that if a protein analyte of 30 kilodalton and antibody response RU is observed for a protein analyte of 30 kilodalton and antibody ligand of 150 kilodalton to be 1150 RU. This is a question. So how much is of the percentage? How much less is of the theoretical Rmax? So that is 82% of the theoretical Rmax. What we are observing Rmax, that is 1150 RU. So during analyte injection, on the general sense of how close the binding response should be observed, binding response of 3%, Rmax 90%, 110%. So if a binding response is greater than was observed, calculation error, incorrect or regarding the stoichiometry of the interaction. So what does it mean? That interaction what we are getting should be less than the theoretical Rmax, cannot be more than the theoretical Rmax. If it is the case that it is more than the theoretical Rmax, then definitely what is happening is that that we have miscalculated the stoichiometry, and that's why it is in the wrong. It is leading to us a false result or false miscalculation of the theoretical ligands. So the thing is that equation can be calculate how much ligand to immobilize on a flow cell to achieve a theoretical Rmax of 100 to 150 IU, ideal for kinetics or affinity measurements. So desired molecular into immobilized into percentage into stoichiometry 30 by 150 into 0.82 into 2 is equal to 457. So now equation can be how much ligand to immobilize on a new flow cell to achieve a theoretical Rmax of 150, 100 to 150 RD. So if we are immobilizing 457 resonance units, it should be getting the 150 RD because we are having the 82% what we are getting. So this is another level of approaching and understanding that how much needs to be immobilized. So use the information generated from your first immobilization section 1 to inform your concentration injection time to get the desired immobilization levels. If 20 microgram per ml for 30 seconds as 5 microliter minute resulted is 400, reduce the concentration to less than 5 in order to not to overshoot the desired immobilization levels. So now what we are doing that we are rechecking the analyte binding and the regeneration. So perform injections of similar analyte concentrations, verify that the bending response is in the expected range, and set up a kinetic titration. So if 120 second injection, 100, set up a titration 150, 25, 10 nanomolar. Titrations run with increasing concentrations. Startup 0 nanomolar analyte. Then 1 nanomolar analyte, 2.5 nanomolar analyte, 5 nanomolar, 10 nanomolar, 25, 50, 100, 5 and 0. So this is how the setup and the experimental condition should look like. So this is what response unit. So illustration shows a uses. One is the baseline, then again association and the sample. So first it is the running buffer, then it's a sample, then it's a running buffer, then it's a regeneration solution for removing the analytes, and finally is again the running buffer. So this is an ideal SPR sensogram where you have the running buffer samples and then going to the association, dissociation, and then finally going to that. So the data from the interaction is presented, providing you with the quantitative data on binding. That's the interactive binder and partner bind the target molecule. Specificity, to what extent does an interacting partner cross-react with the other molecules? How much of a given molecule is present and active? What are the rates of association and dissociation? So this is basically what we know and look upon. So this is another site where you can go ahead and try to find out. So I think the calculation of the resonance units for understanding how much you want to give or how much you want to ligands to be mobilized is what determines the successful experiment, and then you need to have regeneration and then finally uh, the activation. So this allows you to us to perform the analysis and molecular interactions. So this is the target molecules most frequently involved sensor surface, sample containing a potential partner. So this is So these are the ligands, then analytes are getting to it. So quantity, kinetics, affinity. So just see the quantity, kinetics, and affinity. Binding kinetics, how fast or slow does the complex form and decay. Concentration, how much of a given molecule is active. Specificity, how specific is the interaction. So this is the chip which you placed. Right? Angle of minimum intensity reflected light, the angle changes as molecule binds. Okay. So see the ligand, it goes bind to it. Analytes start flowing. Changes, angle changes, intensity, and then again it is going back. So you can see the applications from molecular interaction, from research to drug discovery and QC, development to QC. Yeah, so this is for the guideline, and this was okay. So immobilized ligand, and then 1152. So if you say 100, so okay. Yeah, so this is all for this week, and we will again meet to the next week. Yeah. So, yeah. Thank you all.